In the next section will be Peter Siegel. So as you've heard this morning from Bill Muller, there's a certain connection between Bill and Peter. Peter, in fact, did do his PhD at McMaster University and then moved on to do a postdoctoral fellowship at Memorial Sloan Kettering. Seems to be a place where everyone is congregating, and then basically moved to McGill to become an independent investigator affiliated with many departments, medicine, biochemistry, anatomy, cell biology, oncology, and the Goodman Cancer Research Center. Peter spent a lot of time working on the ERB2 breast model of breast cancer models in Bill's lab, and then went down to the Sloan Kettering to work with Johan Masage and explore metastasis of breast cancer in various models and the sort of effects that TGF-beta signaling might have on the ERB2 signaling pathway. And basically has now sort of branched out and continued this, in the number of metastatic sites possible for breast cancer and colorectal cancer. So Peter, you'll start with the breast cancer models, and then we'll introduce Sébastien. Let's say you guys have about each 25 minutes, 25 to 28 minutes.
Perfect. Great. Well, thank you very much. Can everyone hear me?
Yes.
Perfect. Well, it's been great to be part of the workshop, and I'd like to thank Nicole for organizing this. I've learned quite a bit from our previous speakers. And so what I wanted to talk about, and Sébastien as well, is the utility of patient-derived xenograft models as platforms really for fundamental mechanistic-based research, as well as providing models for translational cancer research. And so what is a patient-derived xenograft model? And so really, in its simplest terms, it's taking a piece of tissue from a patient that has a cancer and implanting that piece of tissue into a recipient model, a mouse model. And this can be a subcutaneous injection or an orthotopic injection, orthotopic meaning placing the tissue in the site which that cancer originally arose. And then growing that piece of tissue out into a tumor or a metastasis, whatever the case may be, and then serially transplanting that tissue from one mouse to the other. And so these PDX models typically are managed and propagated by transplant. So the cells aren't being conditioned to grow on a 2D dish. We keep them as transplants. And this is important in terms of maintaining some of the heterogeneity that these tumor cells have. And so there's a number of things that you can do once you've established a PDX model. And that's shown on the right-hand side. So you can begin to characterize these tumors in more detail in terms of the histology of the tumor. And looking at specific markers that are being expressed by these tumors. And you can compare what's established in the PDX against the original clinical material that was derived from the patient. You can go further and you can begin to bring a lot of the omics technologies that we have available to us in today's day and age in terms of looking at mutational status, whether this be whole genome sequencing or whole exome sequencing, or even targeted sequencing of cancer-associated mutations. Transcriptomic analysis for gene expression profiles. And again, it's interesting to compare the PDX that you've established to the original tumor that came from the patient. But what's more important is maybe the ability to use these models then as tractable model systems to get down to a mechanistic understanding of what's happening with some of these associated mutations and whether they're driver mutations that are really responsible for the aggressiveness of the tumor, or whether they're passenger mutations that really aren't functionally involved in the growth or the spread of these lesions. And so some of these PDXs then can be adopted to organoid culture, and which will allow you then to go in and functionally target specific genes, either by overexpressing them, knocking them down. You can introduce imaging reporters that allow you to follow these tumor cells, how they grow and where they spread within the mouse. And again, if we think about moving to sort of the translational point of the spectrum of cancer research, these are really good models then to assess treatment response to known targets that may have been revealed from your genomics analysis. So you can begin to use therapeutic agents against mutated receptors or oncogenes that are known to be expressed in the tumor. This will allow you to model therapeutic response within your PDXs, or you can model the emergence of therapeutic resistance in these PDXs, and this information might be informative with respect to recurrences that then patients might experience. And so we've heard a lot today about all of the models that you can use to answer questions in oncology and cancer research. And this has run the gamut of genetically modified mouse models or genetically engineered mouse models, the advent of CRISPR-Cas, the idea that you can do somatic gem modeling. And so why is there a need to evoke PDXs as yet another tool that one can use to study cancer progression, cancer growth, and response to treatment? And really the impetus for investing in the establishment of PDX models came from the disappointment in the field with some of the traditional models in terms of how they modeled response to therapy. And so a lot of the labs in the GCRC rely on cancer cell lines that have been established for many, many years, propagated in culture. And you have certain selection that occurs with the establishment of these cell lines. And what the industry was finding was that responses of these cell lines to certain drugs didn't really capitulate the heterogeneity that would be seen in patients. And a lot of drugs and preclinical trials using these as models would look promising, but then they would fail once you got to testing it in a patient population. And so one of the big, and using gem models also really skews the type of tumor that you get to the driving oncogene that you've overexpressed in those animals. And so you're getting less heterogeneity, even though you have some heterogeneity in the gem models. It's considerably less than what you would see in a tumor from a patient. And so the idea was, if we could establish these PDX models, would they be more reflective of tumor heterogeneity that exists within the patient's tumor? And this is what the field has begun to recognize, and that a lot of the PDXs that have been established, whether they be in breast cancer, colorectal cancer, lung cancer, that there's a high concordance of the histological appearance of these PDXs with the tumors that the patient had developed. In many cases, not all, but the sites of metastasis that are experienced in the patient are also recapitulated in the PDX model. And we have a number of examples of that even in our own work. The big part is this degree of tumor cell heterogeneity. And there's been a lot of comparisons done with PDXs and the clinical material from the patient showing that the spectrum of mutations are highly concordant between the PDX and the clinical material. This isn't perfect in that there is a little bit of a selection process going on when you're establishing a PDX. And this has been referred to as bottlenecking, in terms of you may have 15 or 20 different subpopulations within the tumor from the patient, and you may have a selection process where maybe only 5 or 6 of those subpopulations begin to grow out in the PDX. This can be useful because sometimes that selection is indicative of the worst parts of that particular tumor. So there's a selection for the most aggressive subpopulations of that tumor, and it's been correlated with poor outcomes. So those PDXs that have the highest propensity to establish are actually tied to the worst prognosis in the patient. And also some of the molecular features of PDXs that are successfully able to establish look like the genomic features of metastases. So it could be that this selection event is actually picking up the more aggressive subpopulations within the original tumor. Initial passages of the PDX actually retains some human stromal elements, but this is progressively lost following serial transplantation. But the initial passages will have some human stroma elements in there. And more importantly, too, these PDX models are actually been shown to more accurately reflect drug responses that you see in the patient. And so you can begin to take these PDXs out, and depending on the type of cancer that you're looking at, you might be able to model therapeutic response in the PDXs and use that information to devise second-line therapies for the patients in case they actually recur. And this has led to a number of avatar programs where these PDXs that are being established are being used to monitor drug response that then could be informative for patient treatment in the future. And as Sebastian will talk about a little bit in his part of the presentation, you can make organoids from these PDXs that allow you to manipulate genes for functional studies. So like any animal model, there's pluses and minuses. And that's no different with PDXs. And so certainly not all cancers or cancer subtypes can easily be modeled or engrafted into immunocompromised mice. And one of the better examples of this would be in the context of breast cancer, where you can find it difficult to engraft lower grade or stage tumors. They're harder to engraft. And ER positive or luminal type breast cancers have been shown to be notoriously hard to establish in PDXs. Now there's workarounds for this. And it's been shown that you can actually take ER positive cells and introduce them into the ducts through an intraductal injection. And that can enhance the take rate of ER positive PDXs. So there are some tricks to get around this particular issue, but it's not going to work equally well for all cancers all the time. Certain PDXs take a long time to develop. So it can be maybe months and months after the piece is transplanted initially before you actually get a tumor to grow out. And then the time it takes to expand that in order to have enough material to begin to do functional studies, again, makes it a little bit more cumbersome. And this is a model where you're really only looking at tumor growth and metastasis, and you can't look at early events like tumor initiation, which you can do with GEM models. There are some exceptions, and Luke McCaffrey here at the GCRC has been devoting some effort to try to get ductal carcinoma in situ lesions from breast cancer patients and set up PDXs in mice. And in that way, you can model sort of the conversion of DCIS to invasive. But traditionally, these aren't models for studying early steps in the transformation process. But I guess the largest drawback for PDXs is that they have to be established in immunocompromised mice. And so Daniela took us through the concept of immunocompromised mice quite nicely. And so PDXs naturally, they take a lot of money to set up, given the expense of the various immunocompromised backgrounds that one can use. And you really can't model that tumor immune cell interface in PDX models. And again, this depends largely on the immunocompromised background that you're using. However, there may be ways to make these PDX models better with the interest in humanized mice, and we'll hear this in our last talk this afternoon, which should be quite interesting. And another issue is the variability that one sees in how PDX models are being established in different labs. And this is for any number of reasons. One reason is the methods that are used to do the implantation of the tumor cells from the specimen. This could be, if you're dealing with biopsy material as your starting point, surgically resected tumor cells that can be implanted as a fragment of tissue, or if you're dissociating these cells and you're actually injecting dissociated cells. The route of injection is very important. Are you putting these cells subcutaneously and setting up a subcutaneous PDX? Or are you trying to do orthotopic injections to put these tumor cells back in the correct site of origin? And the choice of immunocompromised mice, again, will lead to different outcomes. And I won't go through the table. Daniela did a very nice job. Suffice to say that as you go down the list in the table, you're getting more and more immunocompromised, and this enhances the take rate of your PDXs. It also leads to a potential problem in that you have to be careful that what you've implanted and what grows out is actually a tumor, a solid tumor, or not a lymphoproliferative lesion, right? So the expansion of immune cells that might have come along with the piece of tissue. And so it's been shown that in a number of cases, the lesions that are growing out really aren't solid tumor cells at all, but they're just an expansion of a blast population of immune cells. And so one has to be very, very careful using the appropriate markers to make sure that what you've grown out as a breast cancer PDX or a lung cancer PDX indeed is from the cancer that you're interested in. And so at the Goodman, there's been a number of laboratories that have been quite invested in setting up PDX models, and Morag Park's laboratory really was the first to invest in this approach. She's most known for the PDXs that she's developed for primary breast cancers, and she has a focus on triple negative. And Paul Savage was really the one that spearheaded these efforts in her lab, and they had a nice paper in Communications Biology earlier this year describing that bank of PDXs. She's been active in PDX models for gastric cancer, and this is stuff that she's been doing with investigators at the Montreal General. And I think she's also getting her foot into some lung cancer PDXs with John Spicer and Logan Walsh. As I mentioned, Luke is trying to set up models of DCIS and breast cancer. George Zagopoulos has been heavily focused on PDXs for pancreatic cancer. And I thought that we could take you through our experiences in setting up PDX models from metastases. And so we've been focusing on two different sites of metastases. One being brain metastases, and these are PDXs from numerous solid cancers that commonly metastasize to the brain, which are lung, breast, and melanoma. That should be melanoma. And Seb will take you through our efforts to set up colorectal cancer PDXs that have metastasized to the liver. And so we became interested in brain metastases for a couple reasons. And the first is these are metastases that when they form have a devastating implication for the prognosis of patients. And they're actually increasing in incidence as the systemic disease is better managed. We're seeing a higher incidence of occurrence of brain metastases from multiple types of solid cancers. And when we talk about brain metastases, we need to be cognizant of the fact that there's two main types of metastases that form. The majority of metastases that form are what we call parenchymal metastases. And these are when tumor cells are actually embedded within the brain parenchyma, and they grow out within the tissue. And this accounts for about 80 to 90% of all CNS metastases. And they're associated with poor survival, with median overall survival of less than a year. And then there's a second type of metastases called leptomeningeal metastases. And these are lesions that actually form on the surfaces of the brain in the subarachnoid space. And this is the space that surrounds the brain where the cerebral spinal fluid is contained. And so you can have these small lesions that grow on the surface of the brain, or you can have these leptomeningeal metastases that form on the spine. And this is a much smaller proportion of the total CNS metastasis burden, but they're associated with a very poor prognosis, and they have a median survival of less than 3 months. And so we wanted to get into looking at functional mediators of brain metastasis, and we thought it would be important if we could establish a bank of brain metastases. And I like this slide because it actually shows the integration and the collaboration that needs to be put in place if you're thinking about trying to establish a PDX program. And so it really starts with a strong interaction with a group of surgeons that are willing to work with fundamental scientists to set up a pipeline where this material can be taken from the patient after surgery. That primary tumor material can get banked into a biobank. And this can include bloods, viable frozen material, fresh frozen material, and formalin-fixed paraffin-embedded material from the actual surgical specimen. And we focused on those cancers that had the highest proportion of forming brain metastases, which include lung, breast, and melanoma, although we really were taking all brain metastases when we started the project. And this is really an effort that was spearheaded by an MD PhD student in the lab, Matthew Dankner, who really took this on and put the effort into setting up the PDX program for brain mets. And so what we do is once we've banked some of the surgical resection specimen into our bank, we take a piece of that material and we implant it into the mouse. And depending on the type of tumor we are dealing with, we would put a lung cancer or a melanoma subcutaneously. And if we had a breast cancer, we would put it in the mammary fat pad. We'd let that first implant grow up just to get enough material to then do a serial transplant. And then we would either propagate it sub-Q or in the mammary fat pad, or we would do an intracranial injection and put these metastases back into the brain. And then we would keep those passages going up to passage 3 or 4. And what we began to see was some interesting differences in the growth of these PDXs once we had them in the mice. I should also mention that this was initially started in collaboration with Morag and Paul Savage, who really started the PDX bank that then we took over. And Marie-Christine Guillot is a neuropathologist who's been extremely helpful for these studies. And so this is a bit of an outdated slide, but so far we've been able to successfully, I think it's more like 54 successful engraftments of various different solid cancers into the brain. And so what you can see down here is, depending on the type of the primary cancer that the patient had, we were ranging from 52% engraftment for lung all the way up to a high of 83% engraftment for patients with melanoma. And the ones in green were established by Paul in the Park lab, and the ones in red were established by Matthew in our lab. And what was interesting is when we started to look at these lesions that were growing out in these PDXs, we began to see two dramatically different types of growth patterns or invasive patterns that were being produced. In some of these PDXs, we had what we would call a minimally invasive growth pattern, where we have the brain here, the lesion here, and if we look at a higher magnification image, there's the brain, there's the lesion, and there was a very sharp demarcation between the edge of the lesion and where the brain parenchyma started. And you can see this in a variety of different examples. This was contrasted to this highly invasive growth pattern where you would have the tumor cells growing into the adjacent brain. And in some cases, they were getting fairly far away from the edge of the main lesion. And these cells could invade into the brain in a number of different patterns, either as groups of cells or as individual cells. And we call these this pattern highly invasive. And so when we saw this in the PDXs, we asked the question, is this happening in the patients from which these samples were derived? And what was interesting about this was the field had always thought that metastases, when they formed in the brain, were these very, very well circumscribed lesions that had very little invasion. Unlike a primary glioblastoma, which is highly invasive, surgeons really thought that metastases all were fairly uniformly non-invasive. And this really came from the fact that they looked very circumscribed on an MRI. And so the dogma was that there's not too much invasion going on in these brain metastases. What we saw was actually quite different, in that there was a considerable proportion of these patients that actually were developing highly invasive lesions in the brain. And we could also find patients that had this minimally invasive or more circumscribed type of invasion pattern. And if we took a look at the total number of samples, we had 164 samples that we had a sufficient margin that we could assess invasion pattern. And there's about 66% of them that were exhibiting this highly invasive pattern against 34% that had this more minimally invasive pattern. And as you break it down by the type of solid cancer, breast and lung had a higher proportion of highly invasive to minimally invasive, whereas cancers like melanoma were more of a 50-50 split between the two. And what was important was when we looked at the concordance between our PDX samples and the primary clinical material, we had about a 75% concordance between what the lesion looked like in the patient and what the lesion looked like when we established the PDX. So it was fairly high concordance. And what was also interesting is that we began to see evidence of leptomeningeal metastases in some of these PDXs. And so, as you recall, these are lesions that are forming on the surface of the brain or down the spinal cord. And if we looked at the proportion of PDXs that were forming leptomeningeal metastases, we were able to see that those that had this highly invasive pattern of growth in the brain were the ones that were the most likely to develop these spinal metastases in mice versus animals that were growing minimally invasive lesions. We didn't see evidence of these spinal mets. And if you quantify either just the area of the spinal lesion or the number of spinal metastases, you can see that those PDXs that had a highly invasive pattern of growth were the ones that had the highest degree of involvement in the leptomeninges of the spine. And when we went back to look at the patients, we sort of saw the same thing. So in those PDXs that had a high propensity to form leptomeningeal metastases, the patients either at surgery or post-operatively had a higher proportion of leptomeningeal metastases than those that corresponded to PDXs with low potential to form leptomeningeal metastases, which was quite interesting. So again, these PDXs are faithfully recapitulating the biology that we're seeing in the patients from which they were derived. And importantly, when we took a look at some clinical correlates of these two different types of patterns, we could see that if we look at local recurrence here on the far left, patients that had these minimally invasive lesions had a very low proportion of local recurrence in the brain versus those that were highly invasive that began to have a higher frequency of recurrence. So another growth within the brain around the surgical resection site. This is also correlated into reduced overall survival for patients with highly invasive lesions versus minimally invasive. And again, this correlation that we had with increased incidence of leptomeningeal metastases in patients with highly invasive disease versus minimally invasive disease.
Excuse me, Peter, you have about 2 or 3 minutes left.
Okay, so I'll move through this quickly. Sorry. So what can we do with these PDXs? And so we were really interested in identifying molecular drivers of this invasive program. And in collaboration with Kevin Petrecka at the MNI, we did some single-cell RNA-seq analysis on 2 patients that had highly invasive brain metastases. And the way we set up the experimental design was Kevin took some of the tumor cells that were in the center of the metastases, and he took adjacent brain outside of the contrast-enhancing region on the MRI. And our idea was, so traditionally the surgeon would say, well, there's not going to be very many tumor cells in the surrounding brain because these metastases are so well circumscribed. Our data was suggesting that we should find tumor cells within that surrounding brain because they're a highly invasive growth pattern. And this is what we saw. So we have a melanoma patient and a breast cancer patient that developed brain metastases. If we look at the surrounding brain, we can find evidence of melanoma cells in the surrounding brain, and of course the melanoma cells in the metastasis proper. And the same thing in the breast. These are the tumor cells that are present in the surrounding brain and then in the metastasis proper. And a single cell seq, we're able to identify the cluster of breast cancer cells. Here's the cluster of melanoma cells. But interestingly, you could divide then that breast cancer cluster to those tumor cells that were in the surrounding brain versus in the center of the metastasis, and as well in the melanoma cells, you had melanoma cells in the metastasis center versus melanoma cells in the surrounding brain. And when we compared the expression profiles of what was expressed in these invading cancer cells that had moved into the brain parenchyma, versus the cells that were in the metastasis proper, we found an interesting protein called CRBP, which is an RNA binding protein that's been implicated in cell stress response. And what we were able to show is to validate this actually in both the clinical specimens and in our PDX models, where if we looked at the staining of CRBP, we had a higher degree of staining of CRBP in these highly invasive brain metastases versus minimally invasive in the patient. And we also saw this dichotomy in the PDX samples. And what was intriguing was if you looked at the invading cells at going into the tumor versus the expression of CRBP in the center of the lesion, we saw this differential in expression higher in the invading cells versus those cells that were in the main lesion. And ultimately, we also looked in the leptomeningeal metastases that formed, and we had a higher level of CRBP expression in the spinal nets when you compared it to the center of the lesion in the brain parenchyma. And almost uniformly, there was higher levels in the spinal nets when you compared them to the brain met proper. And so we engineered stable knockdown of this particular target in our highly invasive melanoma PDX model. We chose this model because it was one that had shown evidence of leptomeningeal metastases. And when you knock down this protein, you can see that there's a diminishment in the growth of the tumor cells in the brain. This is by luminescence. This is by staining for vimentin as a marker, tumor cell marker. And you can also see that when we knock down CRBP, the number of lesions in the spine were reduced versus when we had CIRBP present. And then this just shows you the actual tumor burden of these cells when they were in the brain. The degree of knockdown is good, and it was retained over the duration of the experiment. And mice that were injected with melanoma cells that have knocked down for this particular target prolonged the life of these mice when compared to controls. So we've also shown this result in an MDA23 model, showing that if we knock down CRBP, we have a diminished growth in the brain. And so I'll just summarize quickly this part, and then I'll hand it over to Seth. So we have established about 50 PDX models of solid cancer brain metastases. This work really revealed two distinct patterns of growth in the brain, one highly invasive and one minimally invasive. These patterns are important because they are correlated with different clinical outcomes for the patients. And we were able to identify molecular drivers of this invasive program through the use of these PDX models and validate the importance of them for this particular type of pathology. I'd like to say also we've got an interesting PDX that spontaneously metastasizes to the brain. It's a breast cancer PDX. When you put it in the mammary fat pad, you let the tumor grow, we can resect it, and it'll actually spontaneously form brain mets. And we've also pulled CRBP as a driver in that model as well. So we think that this particular protein is quite interesting, and we're working towards trying to figure out what the mRNA targets that this thing binds. So with that, I'm hoping you get a sense of how these models can be used, not only for making a unique resource, but also for getting into some functional mechanistic studies as well. I'll stop there and hand it off to Seth.
Thank you, Peter. Can I ask a little question?
Certainly.
Is it known why the luminal cancers or the ER positive cancers, you have trouble implanting PDXs or growing PDXs?
Yeah, so I think it likely has to do with the fact that the luminals that were coming along were likely of lower grade and stage and were less aggressive. So that impacted their ability to take. But also this notion that where you implant those luminals might have a bigger impact than something like a HER2 positive or triple negative, right, which are more aggressive. So there's been studies showing that if you do intraductal injections of those ER positives, you can increase your take rate, right? So it's a little bit more of a challenging technique, and it takes a lot longer because that natural take rate is sort of limiting the size of the lesions that you can get, but that has been one workaround for luminal type cancers. But for triple negative and HER2s, it can be upwards of 50 to 60% take rate, but luminals it's down below like 25 to 30, right? So it's quite a less effective type of cancer to transplant.
Is this the same thing with every type of cancer? Like if you've got an aggressive lung, it will take a less aggressive.
Yeah.
I don't know if that exists, a less aggressive.
I think that that's true, right? And so there are lots of studies now suggesting that if you implanted 100 PDXs and only 30 of them took, the patients from which those 30 were derived have poor outcomes clinically, right? So clearly you're selecting for very aggressive types of cells within, right, subpopulations within those tumors. So I think that bears out across a number of different solid cancers.
Okay, thank you. So we'll continue on this trend